Hello everyone, welcome to another one of our interviews. Today we're speaking with the authors of the review of structures of Pentatricopeptide Repeat proteins. Today we're welcoming Mark van Raaij the last time as a main editor to the interview. Hello Mark.
Greetings from Spain. Today's a holiday here, but happy to join the interview.
Thank you Mark. Cryst and Anuradha from Brazil. Christy.
Hello, everybody. Nice to see you, John and Mark, again, and welcome, Charlie and you, for the interview.
Right. So, as I said, today we're speaking with the authors of this review. Anuradha.
Hello, everyone. My name is Anuradha Pallakandam, and I work as a research associate at the University of Western Australia.
Thank you very much. And Charlie.
Hello everyone, I'm Charlie Bond and I'm a professor of structural biology at the University of Western Australia.
Thank you very much and welcome to this interview. Right, so the first question is going to be a very classic interview question. What are PPR proteins and why do they matter?
Pentatricopeptide repeat proteins are tandem repeats of 35 amino acids that form a characteristic helix-turn helix structure. And these are abundantly found in plants, and they form a superhelical structure. Upon binding RNA, these proteins are involved in various functions such as RNA editing, splicing, cleavage, and stabilization. So, therefore, these proteins play a crucial role in plant development. That's what makes these proteins very interesting. So. These can be used as programmable tools because of their modular nature.
Thank you, Anu. So can you explain a little bit your research project and why you chose to write this very nice review about it?
My research project was about elucidation of the structure and architecture of S-class PPR proteins and their editing factors. The main focus was to study the conformational dynamics of the S-class PPR proteins and do a comparative analysis with P-class, PLS class proteins as well. So, and this review helped me a lot with the initial framework of comparison, where I could actually study the PPR proteins and compare them with my findings on S-class. So especially with conformational dynamics. So
the reason for the paper also is that we've noticed there are it's often hard to find a historical perspective on a series of structures of proteins. And so, you can find things in the PDB and you can find functional review articles, but things focusing on structure are not as common. And not so long ago, I was discussing with John Helliwell, and he mentioned this particular type of review for that he'd prepared one in Acta a few years ago, and it seemed like a really good home for that style of study.
Well, now with Charlie, my scientific brother, because we're colleagues from Manchester. Charlie, my question for you is regarding the highlights of your review. So in your review, you highlight the PPR proteins. They undergo remarkable large conformational changes of RNA binding, including substantial superhelical compression. To what extent do you think this structural plasticity is functionally required for RNA recognition versus being an emergent property of the repeat architecture?
Thanks, Christy, and it's really nice to be talking to you again. It really is, I think, about the function of RNA binding, because if you think of what the problems are for a protein to try and bind to a long piece of single-stranded RNA, there's a kind of topology problem. You have to wrap the protein around the RNA. And so in order to be able to do that, it actually has to be quite open in order to be able to get on the RNA. And then somehow on the RNA, it's got to find some base pairs that it can bind to, for its recognition sequence. And so then there has to be something that locks it in place. And when the protein's in its more extended conformation, when it's not bound to RNA, the distance between the motifs is too far for interacting with RNA. And so it has to compress so that the right residues can be in the right place to interact with the bases and the phosphate backbone. So that requires that compression.
I must say, I'm fascinated by the modularity of these PPR proteins and how they lend themselves as building blocks in protein design. So, Anu, could you discuss maybe a few examples of modifications or extensions that the community have introduced to these designer PPRs? So, for instance, the desolubilizing helix that you mentioned in the review, that was really interesting.
Sure, John. So the designer PPR proteins are constructed using the repetitive copies of consensus sequences. With the solubilizing helix, they have half a motif of this PPR protein, which was introduced at the C-terminus end of the protein. So it is similar to the solubilizing helix that was found in TPR proteins, and it increases stability and protein folding of the protein. It stabilized these proteins, so it was adopted by many groups because of the effective nature of this helix. So and native PPRs are a bit hard to work with because they're not very stable. So this solubilizing helix actually increase the stability in that way. Some other examples of modifications involve, because of this modular nature, the synthetic PPR proteins, we could modify the fifth and last amino acid PPR code where by altering those PPR codes, we could actually target different RNA sequences. So different groups adopted that, and they introduced PPR proteins which can target different RNA sequences. So like homopolymeric RNA sequences, and they could target native PPR sequences as well by altering this code. So, and another classic example of PPR code alteration was from the crystal structure of 5-ORM. So where they engineered it to bind single-stranded DNA instead of single-stranded RNA. And they showed that these same rules apply for the single-stranded DNA binding as well. This sequence-specific binding capability of designer PPRs was also demonstrated by Shen et al.'s group, who showed interactions between all 4 bases by altering the PPR code. So, and some other examples include the FRET experiments conducted by Mazzano's group. So, where the cysteine residues were altered to bind chemical dyes, and they show the conformational change in the PPR protein. So they saw 50% reduction in the helical pitch upon binding RNA. So they showed significant conformational compaction by using a different technique as well.
Fantastic. Thank you very much.
My next question goes to Charlie. Charlie, while the PPR code has been extremely successful in guiding RNA recognition, and you taught us, all this conformational change in order to do that, your survey also highlights several non-canonical binding modes. How should the field reconcile these deviations when designing programmable PPRs? Are we approaching the limits of a simple recognition code?
Yeah, that's a great question, and really important in the context of the potential functions of PPR proteins, designer PPR proteins. The issue here, I think, is one of evolution. So the modular nature and the application of a simple code works really well for rapid evolution of a protein that can bind a specific sequence. And so in nature, often the target sequences are in mitochondria or chloroplasts, where there's been damage to the genome, which has never been repaired. And so the protein has evolved to recognize the transcript from that damaged gene and to edit it. And so if there's a damage event in the mitochondria in a species, it needs a way to rapidly evolve a way to fix it. And fixing it at the RNA level is one way of doing it. So that explains, why you have a PPR code and something that's rapidly evolvable. The question about the non-canonical binding modes, I think, comes to the fact that PPR proteins then have to specialize in some cases. So if you've got an important transcript that needs to be bound, you rapidly evolve a protein that binds to it. But then you really want to make sure the affinity is really high, and that it's very selective for that sequence and not other sequences. And so many of the examples which show these non-canonical binding modes are transcripts where the job of the PPR protein is simply to bind to the transcript. It binds to it, and that stops nucleases being able to attack the transcript. So its only job is to bind as tightly as possible. So you can think of it maybe as like a maturation step. You evolve a protein that can bind the transcript, and then it gradually evolves to bind very, very tightly and very specifically. And it's that step of evolution that introduces all these idiosyncrasies and special individual interactions. So what that means for protein design is that there probably is a limit on how high an affinity we might be able to engineer, and maybe even how high a selectivity, with it just within the sort of simple code.
Okay, so my next question now goes to Anuradha. Anu, given that AlphaFold captures the helical architecture well, but it struggles with conformational states and RNA positioning, how do you envision integrating experimental structure methods and AI predictions for studying highly dynamic systems like PPR proteins?
That's a very nice question, Christy. So the AlphaFold is very good at predicting the helical architecture of PPR proteins. So it predicts them with higher confidence. So one of the problems that I noticed was that when we compare the apoform of PPR proteins, they actually have 10 repeats per superhelical turn, whereas the AlphaFold predicts it as a RNA bound structure, which shows 9 repeats per superhelical turn. So there are some discrepancies in the prediction when it comes to the conformation of these proteins. But we can still, because they predict the helical architecture with high confidence, we can still use them as initial models when we are building models into our SAGS data or cryo-electron microscopy or crystallographic data. And as the new structures emerge, I think the unique conformational states of these proteins in their bound and unbound conformations can be predicted much more accurately because of the availability of the data, which can be used to generate better models, and we can get accurate structures. So from that, so that's what I think. So currently we can use them as an initial model. So, but we can't be very confident in the low confidence regions. So,
moving on to a different subject, Anu and Charlie, if you would like to answer this as well. How is your research team organized?
I'm very, very glad to be in Charlie's team. So he's an excellent supervisor. So very supportive, very knowledgeable, and like we have wonderful postdocs in the lab as well. So though we have individual projects, we all work as a team. So especially when it comes to troubleshooting. So, and it's very pleasant environment to work with. So, and because Charlie's lab is structural biology lab, so we collaborate with other labs like Ian Small, who does more work on plants and like relate plants, basically like PPR proteins. They do a lot of biochemical and genetical modifications, and they learn more about these proteins. So, and they do a lot of bioinformatics as well. That collaboration is very amazing. So I'm very fortunate to be in his lab. So.
And I'm fortunate to have Anu in the lab. Because of her collaborative nature. So I really like having strong collaborations with people where our interests overlap, but not too much and not too little. And it works really well with Ian Small. And so I'm very lucky to have other biological collaborators who are the same, where we understand enough of what each other do that we can work well together, but we don't have conflict with each other. And by having Anu comes to my lab meetings, but also goes to Ian's lab meetings, and so we kind of share people around so that they get a very broad biological and structural view of whatever their project is.
Last Friday, when I was talking with John and Christy about organizing the interview, I got shown on YouTube a geographical curiosity claiming that Perth is the most isolated city in the world. So, both to Anu and Charlie, how does this impact your life and your work?
Perth to me is my home, so it's very beautiful, and it's actually one of the sunniest cities, not only in Australia but in the world as well. So, and it has quite a good community sense. So, and people are very friendly. So, and we always like that nature. So wherever we go, they smile and greet us with a warm welcome. So, and yeah, and it's very, like, people collaborate a lot. So it's very helpful in the scientific field as well.
I understand it's quite a big city, no?
Yes.
Yeah, 2 million people. Yeah. Yeah, so the thing about isolation is a bit relative. I used to live in Dundee in Scotland, and people used to think that was a long way away. And living in Perth, you just develop a different perspective. We'll drive 4 hours, 5 hours to get somewhere without thinking too much, maybe for the weekend. We get used to having to fly to Melbourne. I might go to Melbourne or Sydney or Brisbane for a day or two for a conference, but the distance from Perth to Brisbane is about the same as from London to Tehran. But culturally for us, it's just like going from York to London. So I've now lived here 19 years and I love it. It's a fantastic place to live, and I thoroughly encourage anyone to come and visit us.
Okay, thank you very much.
Do you have any questions for us?
I've maybe got a question going back to my comment about why we submitted the review to Acta F, and what do we need in terms of review information, articles out there to guide us through this world of protein structure that's expanding massively and diversely currently with all of the new computational tools? How do we keep a handle on it? And are topical reviews like this the way to do it?
I'm quite happy to answer yes. I think they're massively useful.
Yes.
There is so much information that just remains undigested, and it's so hard to get a hold of. I think having a an expert hand guide you in different subjects is absolutely invaluable, and I would like to invite other people to submit reviews like yours, because it really helps.
One of the things I found satisfying in when we were putting this together was to be able to put a bit of a historical perspective, because you lose that. You can't get that from a database. And so sometimes the order in which discoveries were made is actually an important part of the story. And that's something you often get when you see a great scientist speak, and you get the narrative of their work, whereas before you just knew they had 200 papers.
Yeah, what I was gonna say is we strongly support this kind of reviews, in particular for students who are starting their theses. So it's a good way for the students also to have a broader, idea what the project will be. So we wrote a editorial about it. So we should have more papers like this. And a lot of contribution from students and postdocs.
Okay, so we've come to the end of the interview. First of all, I would like to thank Mark van Raaij for all those years of service to the journal and for his participation in all these interviews that we've done. I think this is the 10th interview that we do. So thank you very much, Mark. And thank you to the authors of the review, Anu and Charlie, and hope to see you soon.